Good day everyone. So today we are presenting our lab report entitled Mitosis. An introduction of observing the stages of mitosis with microscope. These are the members: Bisactricia P, Cantillo Carlin C, Hayag John Peter C, Marvida Marian S, Payong Ellen Mea T, Reyes Mariah Kisha DB. So this is our objectives. Identify the stages of mitosis. Compare the shape and structure.
So for our introduction, in this activity, we identify the basic parts of the specimen, namely onion roots, Allium cepa. This activity aimed to help us observe the structure of plant cell, the process of mitosis, as well as the appearance under the microscope.
For our materials, we used glass slide, cover slide, iodine solution, carmine aqueous solution, medicinal drop, dissecting kit, and microscope. And the specimen we use is onion roots. For our methods, first we prepare the needed materials and the onion root. Second, we cut a small portion of the onion root to be observed under the microscope. Then, we place the cut portion on the glass slide and we add a drop of iodine solution, also carmine aqueous solution. We cover it with cover slip and observe it under the microscope. And lastly, we identified the different stages of mitosis.
Result and discussion.
We used iodine solution as a staining liquid. We can clearly see the compact structure of onion and we can see the cell, but we cannot identify if it is undergoing mitosis, since we can clearly see the nucleus.
Onion root using carmine acetate solution. In the onion root stained with carmine acetate solution, the low power view shows a noticeable red or pink coloration which is much stronger than the iodine stain. This stain highlights regions where the cells are dividing. Under high power, the structure are much clearer. You can see the cell walls, cytoplasm, nucleus, and even stages of mitosis are distinctly. The cells take on dark red to pink colors, making the internal structure easier to identify. This stain provides higher contrast and details than iodine.
In comparison, our microscope view of the onion root tip showed the basic features of mitosis, but the details were not very sharp. We could see dividing cells, but the chromosomes and stages were harder to distinguish. In contrast, the online reference image is much clearer, with well-defined chromosomes and easily identifiable mitotic stage. Both images show the same process, but the reference provides the detailed view that supports and clarifies our actual observation.
In conclusion, in this laboratory, we observed the structure of plant cell and the process of mitosis. We use a specimen like onion to study different cell parts under the microscope. The specimen shows unique shape and features. We also examine onion roots, tips to identify the different stages of mitosis. Overall, the experiment helped us understand how plant cell function and how mitosis support plants' growth and development. Lastly, this is our documentation.